what I'm going to focus is on mostly on the neuro transplantation cell therapy aspect. But before that, I want to disclose that I'm the co-founder of Brain Cell in medicine and also the Brain Cell Therapeutics actually in San Diego. Okay, so because I'm going to focus on the nervous system, I think everybody here or in Zoom knows that the brain actually is built precisely by numerous of neurons and glial cells. And I think if we Google it, there will be like 80 to 100 billion neurons. What I would like to emphasize is that these neurons are all specialized. There are many, many different kinds of neurons that build this kind of network. And each neuron, have all this connection. Again, it's a specialized connection called synapses. And that's why our brain operates very much like a network of this numerous specialized network, and it's like a supercomputer. Of course, way more than just a supercomputer. Any computer in the world cannot compete with the brain we have. So that actually brings to the question when it comes to repairing the brain, the damaged brain or diseased brain is very, very tough because we not only need to replace the lost cells, but also need to rebuild or at least modify the neural circuits in order finally to restore the function. So it's very challenging when it comes to repair the nervous system. Now, there are many ways to repair the brain, right? One is to have the stem cell in the brain to regenerate. But of course, that has been explored by many people here in San Diego. And the issue is that those stem cells are generally limited in certain area of the brain. And so when it comes to other parts of the brain, it becomes difficult. An alternative way is to turn some of the cells, let's say glial cells, into neurons. So that's a very exciting area. I think it's a potentially very promising area of regeneration. And the slightly better developed area is cell transplantation, meaning transplanting exogenous cells to replace and repair the lost neurons. And this will work better, particularly for some diseases with localized damage and particularly certain cell types. And ideally also the circuit is relatively simple. So this will be a better option for cell transplantation strategy. And a good example perhaps is Parkinson's disease. So Parkinson's disease is caused by degeneration of the dopamine nerve cells in the midbrain, particularly substantia nigra. And when these neurons are degenerated, so they usually connect with the so-called striatum, caudate putamen, and they release less dopamine, and so it leads to a series of symptoms related to movement disorder, like chamber rigidity, postural issues. And so because it's a well-defined, relatively well-defined disease based on the cell type and the mechanism. And so now we have a reasonably good treatment. For example, we can use L-DOPA to replace the lost dopamine, or use surgical to modify the circuit, which I mentioned. The problem is this treatment usually do not last very long. Let's say L-DOPA treatment after several years, when the dopamine neurons keep dying and lost, they cannot convert this L-DOPA to dopamine. Then you need to keep adding large amount of L-DOPA in order to work. And in the end, they usually lose its effect. And so that's why people are trying hard to develop alternative ways to deal with the debilitating disease. And cell therapy is one of the potential treatment that people are developing. And this actually has been tried for quite a long time. It's surprising, initially using fetal tissues, and particularly by the group in Lund, Sweden. I think when they dissected out the midbrain tissues, usually 5 to 9 weeks old embryo, and take quite a few embryo, the tissue from a few embryo, and then transplant into one patient. And for some patients, actually, they live for quite many years after transplantation. They were later on less dependent on L-DOPA. Their symptoms improved quite substantially. But in the double-blind clinical trials, actually there were 2 in the US sponsored by NIH. In those 2 trials, they were not very successful. They were very much mixed kind of outcome. And so when many people look at the issue, why some succeeded, some failed, and there are many, many issues involved in a clinical trial. But one of the main issues is the inconsistent and the poor quality of the donor tissues. Because you need to collect quite a few tissues together and wait until you have enough to put into one patient, and that's a tough job, And so the hope is that if we have the consistent source, cell therapy might work for Parkinson's disease. And that brings the hope when the pluripotent stem cell was first established by Jimmy Thompson. It's called embryonic stem cells, and later on developed further by Yamanaka, the induced pluripotent stem cell. Because those cells can give rise to pretty much any kinds of cells that build our body, including nerve cells. So initially, we took some effort to generate this human embryonic stem cell to dopamine neurons. At that time, because we knew that the Wnt is important, but we could not really activate the Wnt effectively, so we used FGF8, which is also involved in midbrain dopaminergic development. So at that time, we generate dopamine neurons and put into the rats lesion with 6-hydroxy dopamine that creates the Parkinson's model. And in this model, we can actually repair replace the lost cell and made the animal recover from some of the motor deficit. But those neurons may not be the right type of dopamine neurons because of the our ability to really pattern to the midbrain. So several years later, I think quite a few labs almost simultaneously developed the method to guide the human stem cell to really authentic midbrain dopaminergic neuron because of the availability of a small molecule called CHIR, which can pattern the cells very precisely to the midbrain. And from Lawrence's lab, Martin Palmer's lab, and our lab, we kind of guide the stem cell to dopamine neurons. And this actually is reasonably simple. So if we give the stem cell, when we guide the stem cell to neuroepithelial cell, and at the same time, give the small molecule to activate the Wnt signaling, and you can see the dose-dependent change in terms of transcription factors that relate to the anterior to posterior. So at one concentration, roughly 0.6 micromolar, we can see that the cells turn into midbrain type of cells. And of course, the higher concentration will drive the cell to hindbrain and cervical spinal cord. And then in the presence of a hedgehog, which needs to be ventralize the cell to the floor plate, because the midbrain dopaminergic neurons come from the floor plate cells. And so in the presence of both of these factors, these cells, many of the cells become dopaminergic neuron express tyrosine hydroxylase, as well as a whole series of transcription factors like INGLIO1, FOXA2, LMX1A, and so on and so forth. Interestingly, when those cells are differentiated in a petri dish and using this method, they actually also behave like the neurons in vivo. They have a tonic firing kind of properties. They even have this sag potential, which is characteristic of the dopamine neurons in the substantial nigra. And so we have those cells. Then the question is, can we really repair the brain? So there are a series of issues we need to really address before we really move toward that direction. One is that whether we need a very specific type of neuron for each of the neurological conditions. Probably the more important question is, if you put the neurons into the brain, whether they can project the nerves to their right target. The equally important question is whether the transplanted neuron also receive inputs, because the neuron connect to each other in pre- and postsynaptic ways so that you can regulate the neuronal activity in a proper way. And so finally, of course, whether that kind of connectivity really contributes to the functional way which is lost in the particular disease. And so the first question is, for Parkinson's disease, when we transplant dopamine neuron, how do they work? So the way we addressed this question was to use optogenetics and chemogenetics so that you can regulate the neuronal activity after they were transplanted into the brain. So I think Lawrence Studer's lab used optogenetics, and our lab used chemogenetics. So basically, we build genetically modified the cells, engineer the switch, functional switch. You can turn on and off the function of the cell when the cells are transplanted into the striatum. So in this case, we both transplant the cell into the striatum. So the whole striatum is filled with the human axons because when they transplanted dopaminergic neurons, the cell will fill the gap. And then we turn on and off the function of the transplanted cell and look for the activity change using electrophysiologist recording of the brain slides. And just to summarize, it turns out that the grafted human dopamine neurons, the way by which they work is to regulate the endogenous signaling from glutamine signaling to the GABAergic neuron in the striatum. Basically, what it says is that the way the transplanted cells do is very much similar to the way the endogenous dopamine neurons work to regulate the signaling between the glutamate neuron from cortex, thalamus, and elsewhere to the striatum. That's how it modulates the motor activity of the neural network controlling motor activity. Now, the transplantation of dopamine neuron into the striatum mainly tests whether the cells produce dopamine or not. It really does not address some of the questions I initially put. One is whether they can project and find their target. Two is whether they can receive the appropriate signals. And so what we did is transplanting the cells back to the substantial nigra. That's where the dopamine neurons live normally. And when we transplant the cells there, we can ask a question, can they glue their axons? Where do they glue? Can they find their right partners? And so just to show you a couple of slides, here is a sagittal section, serial section. And you can see this is the transplanted area. It's the substantial nigra. And the dark dots or fibers are the human-specific labeling for human nerves. And you can see these fibers go from the nigra all the way to the striatum. This is a magnified view, and you can see in the striatum. So meaning, one, the axon actually can project in a long distance. Two, they actually go into the striatum. That's where the axons, the midbrain dopaminergic neuron, particularly the substantial nigra dopaminergic neuron, normally go. And to make it a little bit further, if you pay attention to here, the fibers are actually mostly in the dorsal part of the striatum. If some of you know the neuroanatomy, the midbrain dopaminergic neuron in the substantial nigra project mostly into the dorsal striatum. Those in the VTA project into other areas, ventral striatum and other parts of the brain relate to rewarding other kind of function. And so it's striking that the cells project exactly to the dorsal striatum. That's shocking to me initially when I saw their slide. And we use, this is a cross section. It's the same, just to show you. It's using a different method. Use TH reporter, and you can see the tyrosine hydrolysis fibers are mostly in the striatum, again, in the dorsal striatum. So the cells can move and find their target. And is that the question then is, how do the cells do this kind of job? Or if you place any cell type into the substantial nigroid, they will do the same job. And so we actually transplant a different type of cells. One is dopaminergic neuron, another is, so the dopaminergic neuron, I just show you that they project to the dorsal striatum. And we also differentiate the cortical neurons from the same stem cell and transplant to the same place, nigroid. And now you see the cell also can project, but they project elsewhere. It's thalamus and the cortex, other areas, which are the areas that the cortical neuron project to. So this set of comparison means that the cells can project in the mature brain, but where they go depends on the cell types. They have their cognate partners, and they will find them, and that's striking, Another part is, how do they achieve that goal? If we zoom in and look at the cells, dopamine neuron transplant into the substantial nigroid and follow the fibers, how they get to the striatum. And you see that they actually follow the midbrain, forebrain bundles. That's the way normally the dopamine neurons in the substantial nigroid go in vivo. And once the fibers get to the ventral part of the striatum and then turn around, and they're going up to the dorsal part. And then they branch out extensively. And this route is exactly what the endogenous dopamine neurons do. And that's how striking it is. And this, of course, I show you just as an example for how cells find their targets. The reality is that we actually did a series of work over the past decade that the cells always behave like that. For example, almost a decade ago, we transplanted the medial spiny GABAergic neuron versus GABAergic interneuron into the striatum for Huntington's disease. And it turns out that only the medium spiny GABAergic neuron can project to their targets and correct the functional deficit, not the interneuron. Even though they both are GABAergic neuron, even though both can survive, but only the medium spiny GABAergic neuron. The similar thing is when we look at the medial septum lesion, create the learning memory model, deficit model, and then we use GABA interneuron and the cholinergic neuron. One is forebrain, another is spinal cord, meaning different type of the same kind of different type of neurons with same type of transmitters. But again, it's the forebrain cholinergic neuron and the GABAergic neuron connect to their target and contribute to the function. And even in the spinal cord injury model, I think this is what I collaborated with Mark Telinsky here. When we place the forebrain neuroprogenitor cells versus spinal neuroprogenitor cells, and it's the spinal progenitor cell that attract the corticospinal tract into the graft. We actually recently did that because now we can actually generate different subtypes of spinal interneurons. And we transplant to the spinal cord injury model, and they find their specific targets. And so this kind of cell type specific kind of path finding their partners and contributing to their function is really quite a general phenomenon in the way that in the past we felt that the neuron usually cannot do that in vivo, but they actually follow, even in the mature brain, the immature neurons still behave like what they do during development, and which opens the possibility for cell therapy to work, I think. So I talked so much about where the axons go, how they go. The other side of the question is, after the cells are transplanted into the brain, do they receive the right inputs? And that is very important, because if the transplant cells do not receive the correct inputs, their transmitter release, their circuit activity transmission will not be regulated appropriately, and that can create problems. And so the way we did was that we labeled the cells genetically, and then we can use rabies virus tracing to look at what presynaptic inputs the transplanted cell received. And we also labeled the cell using reporter lines, let's say tyrosine hydroxylates. And then we can take the brain and slice them and electrophysiologically record them to look at what kinds of input they receive. Because this, and we compare the inputs they receive when we transplant cell into the substantial nigro versus the striatum, and to ask the question whether they receive the similar inputs, what dictates the input that they receive. And just to summarize, because it's a huge amount of data, if we look at the rabies virus tracing, and we look at where meaning which part of the brain, which parts of the brain they send exons to the transplanted cell, and we compare with endogenous dopaminergic neuron, it turns out they are quite similar. However, depending on where we transplant, meaning striatum or the substantial nigro, the transplantation site can influence the input. And this is important. But then we look at the electrophysiology recording, look at the functional inputs, because this is the most important part, because that will tell us how the activity is of the transplanted cells is regulated. And the functional inputs actually is established long after the anatomic connection, meaning we actually observe this anatomic connection based on rabies virus tracing in just a few months after transplantation. But the reality is there is not much activity. If we look at postsynaptic current, it takes at least 6 months or longer in order for them to establish functional connectivity. And that's important because that can explain why when we transplant the cells already make connection, the reality is we do not see much improvement because it takes time for them to establish functional connection. Another part is what kind of inputs. In this case, we just categorize in a very simple way, excitatory versus inhibitory. And then if we compare different type of neurons. Red is dopaminergic neuron and green is non-dopaminergic, just in a simple way. And then in the striatum versus the nigro, and compare them. And it was just to summarize, because it's a very complicated way, just to tell that in this way, if we only look at the inhibitory versus excitatory inputs, if we compare dopaminergic neuron versus non-dopaminergic neuron, and so the dopaminergic neuron usually receive more this kind of inhibitory inputs than the excitatory inputs. And if we compare the dopaminergic neuron we transplanted versus the dopaminergic neuron in situ, meaning those in the substantial nigro, they are very similar actually. And that piece of data tells us that, hey, the cell type, again, can dictate the inputs they receive if we put back into the right place. So that's even more exciting, meaning if we have the right type of cells, they not only can project out to the right place, target, but also receive the appropriate signal. And so that will make the cell therapy work. And that actually is illustrated here by looking at the functional outcome. We use a number of different behavior measures for the animals. And one is the rotation test. So normally, when we transplant back to the substantial nigro, you can see the rotation, because we only lesion one side of the striatum nigro. So when we stimulate the animal with amphetamine, they release dopamine, and so they rotated to one side. When after transplantation, particularly transplant to the nigro, they recovered to the normal baseline. If we transplant to the striatum, they recover much faster because you put it right into the target area. But often they overcompensated because those cells are less regulated, well regulated, because it's in the wrong place. So the video simply want to show you that we can actually regulate the activity of the neurons, and so you can regulate the behavior. So I just want to summarize here. So far, what I have shown you that we can generate the right type of cell. I use dopamine neurons as an example. Our lab actually has generated many different kinds of neurons. But as I mentioned, that this dopaminergic neuron have the right transcriptional code, and they have very characteristic, even the physiological behaviors. The important part I mentioned that when we transplant those cells, they actually know where to project and who to find as a target, and also receive the right inputs. And the important part in this building of the neural circuit is that it's the path finding, the target finding, and the partner, the inputs partner, to a large degree depends on the cell identity. And this is a very interesting finding, and once they rebuild this kind of pre and postsynaptic network, and you can see this functional recovery in those mice. With this kind of finding, we move on to the preclinical model in non-human primates. So one way we did was to take the skin cell from Parkinson's monkey and differentiate them into dopamine neurons and then transplant back to the monkey. And this is autologous transplantation. We set up a control that is transplanting to another monkey, which would be allogenic transplantation. And in this case, we pick up relatively old animals because Parkinson's disease developed relatively in late stage of our life. We made the cells and let made the Parkinson's model and let them stay stabilized for a couple of years, because normally those, if we are going to do cell therapy, those patients probably will not be willing to do that initially because they have a reasonable treatment option. And so we actually try to mimic as much as we can to the clinical condition. So we waited quite a couple of years or a few years and before we do transplantation. Because this is to test for autologous transplantation, so we did not give any immunosuppression for both groups. And then look at their behavior and imaging and so on and so forth. And it was interesting to observe that pretty much the animal began to show symptom improvement starting from roughly half year. The blue dots are the animals with autologous transplantation. The red dots are the allogeneic transplantation. And you see both the clinical rating score and the fine motor skill tests, both shows begin to recover at about 6 months post-transplantation and relatively stabilized after 1 year. And so pretty much all those animals recover in this way. And correspondingly, when we use PET imaging to show the dopamine binding potential, you see that in the allogeneic transplantation, you do not see much. This is the lesion side. And then this is autologous animals before the transplantation. You do not see it. That side is pretty much empty. In this after transplantation, now you see this binding potential increased substantially in these sites. And so corresponding to the behavior improvement, we see this dopamine release and binding here. And then later on, when we look at the histology, you see even in the allogeneic transplantation, there are grafts. Actually, even without immune suppression, the grafts are there. But usually in the allogenic transplantation, they are small and they are compact and they have a clear boundary. And this is a typical autologous transplant. You see the graft is bigger but more diffuse. You do not see boundary. It's almost like a fuse to the host tissue. And this is slightly higher magnification. You see the axonal growth. These are TH fibers grow out. And you see the axonal terminal branching out. And these are the typical morphology of the endogenous dopamine neurons in the substantia nigra. And this is exciting because we have several animals. We look at the relationship between the number of dopaminergic neurons in each animal, the amount of PET binding, and the degree of behavioral recovery. It turns out there is a correlationship between the number of dopaminergic neurons in the transplanted area and the behavioral recovery. And roughly, we feel that it requires roughly 70,000 dopaminergic neurons for the monkey brain in order for the animal to recover. And we roughly calculated that the monkey requires that. In human, it's roughly 4 times bigger, and it probably will need roughly 300,000 dopaminergic neurons for the patients. And we look back what has been shown in the past with fetus tissue transplantation, where the transplant was successful. It turns out that roughly in this range when those patients show recovery, and there are roughly this number of dopamine neurons in the brain. Okay, so this piece of preclinical study showing us that the autologous transplantation, it works even without immunosuppression. It at least make the animal recover. But the allogeneic transplantation will be variable. It tells us that if we are going for allogeneic transplantation, we will need immune suppression, because otherwise the tissue probably will be limited. And there is a clear correlation between the number of transplanted cells with the behavior and the PET binding. So I used 2 examples, one in rodent, one in monkey, to show how we start from the stem cell and gradually move toward the clinical directions. And I mentioned earlier that in the 80s, and people already started trying using fetal tissue for treating Parkinson's disease. Now I think we have a number of groups working on it. One actually was done by the Harvard group using autologous cell therapy that was published in New England Journal of Medicine. I think now there are several groups around the world working on. Some are already in clinical trials. So we are on the way. Of course, there are a number of issues still need to be solved. I think I only use maybe the dopamine neuron as an example. We feel that we are ready, but there are still rooms for improvement, I think. For example, the dopamine neuron we generated midbrain type of dopaminergic neuron, but may be contaminated by some other kind of neuron, because when we use one concentration of a morphogen, we usually ended up with a spectrum of the cells besides the main target cells. Whether we use high or low wind agonist, we will end up with the same things. And so we need to really work out. This is just the example for dopamine neurons, but for other neuronal type, it's the same. So the principle we put forth in our review article, I think in 2016, is you can actually limit the cell to only one type of cell by restricting the cell to only one tiny brain regions. Another part, of course, related to cell therapy is whether we go after autologous or allogeneic. There are always debate in terms of science, in terms of business or cost, all sorts of things. I think this is something we may deal with each of the disease in a different way. So far, I talked about rodents and now monkey, but still they are animals, they are smaller. When it comes to human, a simple thing is the size and distance. For example, if we are going to treat stroke patients and we transplant the cell into the cortex, in mice, it's still pretty long, but it's doable. But when it comes to human, and if you want the neuron to grow from the cortex up down all the way to the brainstem and the spinal cord. It's a daunting job, and so we still have a long way to go, I think. So I will stop here by thanking a number of people who did involved in this work, particularly Man and Yue-Jun both left my lab several years ago. I think they have their own lab. I think they even got tenure, I think. Yunlong is the one working on the monkey transplant. He's in the market now for faculty job. And I want to thank my longtime collaborator, particularly Melina Emborg, for working with me on the monkey studies, and Brad Christian for the imaging part. Of course, for the funding source, particularly NIH. And thank you. I will stop here for any possible questions and comments.
All right. My question is first about the safety issues. I'm assuming that none of those experiments, even in the monkeys, you see any kind of tumor arising from those cells. And imagining in humans that this would be there for long times, I mean, how can we be sure that these cells will not develop something that is not what you expect?
Yeah. This always the first thing when we move on to patients, right? Safety is number one. You have to get through FDA requirement. I think in most of the animal studies we have done in our lab, and also because we are involved in a consortium, international consortium on Parkinson's therapy. I think so far the animal work, both in rodent and monkeys, show that these cells are safe. Safe in a way that because we differentiate the cell to neural, you've been working on neural differentiation, even though you work on mostly organoid, but still, after 2 weeks, it's very hard to imagine for the stem cell, pluripotent stem cell I'm talking about, to survive under that kind of condition. That's number one. Number two, that the current neural differentiation, particularly for dopamine differentiation, is quite efficient, and the cells are mostly in the neural lineage. Not even much in other meso or endoderm lineage. And so in a way that it's quite limited for the cell to grow as tumor. Of course, when it comes to moving toward the clinic, there will be a lot of assays required by FDA to demonstrate that they will not form tumor, both in terms of in vitro QC, quality control kind of measure, as well as in vivo safety measures. I think that is the way we do for every drug, every new drug, I think we will have to deal with.
So great. So you have to realize this is not coming from a neurologist. So with your last point about the anatomy of where you transplant these cells, and I didn't sort of fully appreciate that you were putting these in the cortex and they were migrating or whatever to this substantia nigra. But I mean, I actually remember those studies by Kurt Fried, 20 years ago, where they actually, drilled in the brain, to be blunt, right, and implanted them, I thought in or near the substantia nigra. And so I don't know, like you said, there are some studies going on, I think, Lauren Studer's group and others. Where are they implanting them? And then the second question is about, the underlying cause of the Parkinson's disease, right? So unlike the animal models, you're putting them into, some sort of environment that I don't even, think we know what causes the disease.
Okay, I try to understand. So the first question is where to put the cells in which part of the brain. I mean, so mostly in clinical trial for fetal tissues in the past, And for the clinical trial now ongoing, or many of the clinical trials are in the planning, the cells are put into the putamen or chordator, mostly putamen, or we call it the striatum in the brain. So those are the area where the dopamine nerve cells project their axons to, so meaning their target area. Because if we transplant into the substantial nigro in the midbrain, the nerve needs to grow in a long distance to their target, which would take probably years. And so that's why for clinical practice, we want to overcome that distance barrier, just directly put into their target area. So that's what I mentioned, that there are issues involved in putting into their target versus in their home location, because their inputs, the cell receive to regulate their transmit release is different. So that's one question. I think your second question, if I understand correctly, is that the model we use actually is an acute kind of lesion. But the reality in Parkinson's patients, there are a lot of pathology going on, right? And this pathology could invade or make the transplanted cell undergo the same type of pathology. Is that what I guess that's what you are alluding to. And indeed, there are evidences to show that the transplanted cell may also develop this kind of degeneration type of pathology over the years. However, it takes time, just like a Parkinson's patient develop the disease, over 40, 50, 60 years. The same happens to the transplanted cells. I think there are 3 or 4 reports when they look at the fetal tissue transplantation after, I think 11 years, probably is about 2% of the cell showing some pathology. I think 24 years, maybe 11% of the cell showing this pathology. And normally, we usually lose 80% of the cells before we show symptoms. So in a way, even they are going to develop this kind of pathology, it's still a useful therapy because it takes many, many years for them to develop.
Let's take a few of the questions that are coming in online. One of the questions is kind of pertinent to what you were just talking about. The questioner said that he loved the autologous approach, meaning iPS-derived cells from the recipient. But this questioner was asking, for those that believe that Parkinson's disease has an autoimmune component, would that be a problem in that you're actually taking cells from a patient that may have derived the disease because the system was attacking the dopaminergic neurons or others?
I think that's a great question. If it's autoimmune disease, then you have the problem because you have the same target cells. I don't know how strong the evidence is about autoimmune disease. Let's say there is one. I think ideally, we probably should modify the cells before we do transplantation. That probably would be ideal. I think even, I don't know how strong the evidence, because even for autoimmune disease, those patients still develop the disease usually in their decades of life, right? So hopefully, if we can still help the patient for 2 or 3 decades, I think it may still be useful. But I do agree that if there is autoimmune components, I think ideally we should modify the cells to evade the immune reaction. Probably would be a better way to go.
This questioner was asking, I'm combining a few of the questions now, This questioner was asking if you could clarify what immunosuppression did with the allograft animals, and was wondering, a few of these questioners were wondering, when you omit immunosuppression from the allograft, does that mean the allograft does not work as well as the autograft? And then another question would be, if you did use immunosuppression with the allograft, would those cells now function just as well? And that might be a practical therapy.
Okay. So in the experiment we did for the monkeys, we actually did not use any immune suppression, because I mentioned that we tried to test the autologous transplantation. So under that kind of condition, the allograft, they are still graft, but I mentioned that they are small and they are restricted. So that's why I was saying that ideally, if we are going to do allograft, we should use immune suppression. And I think people did, particularly Jun Takahashi's lab from Japan, I think they did this type of work. If the cells, the transplanted animals are under immune suppression, the allograft can survive and contribute to the functional recovery.
Okay, so you think that had you used immunosuppression, then those monkeys might have done as well as the autograft.
Correct.
Which then gets into another question is, in a very practical way, when you need to start treating patients, is your intent to make IPS lines from every patient, or do you think you could have a universal donor line, maybe with some immunosuppression?
Right. I think for autologous transplantation, which we are doing now, we will make iPS cell from individual patient. In this case, we don't need immune suppression because we already shown in the non-human primate we don't need immune suppression. However, if we are going to use one cell line, whether it's from EA cell or iPS cell, and apply it for all the patients, which is allogeneic transplantation, then we need immune suppression. Of course, another part is that many people are now working so hard trying to produce the hyper-immunogenic cells or the universal donor cells. That will be wonderful if they can work even without immune suppression. Probably idea at the beginning, we may still want to do some immune suppression to protect the cells first, and then taper off.
Great. Any questions out in the audience? Okay.
Going back to that question about the site of the transplantation, because your data suggests something else that you are trying to do clinically. And I understand, I mean, you're imagining that would take several years for the right projections to be in the right place. But once they are there, I think they would work much better than if you transplant right on target. And a related question, which I think it's amazing biological question, it is, you are transplanting very young embryonic cells. How come they're finding the right pathway in a more mature brain?
Terrific question. The first question, ideally, I would love to do that. And in that case, you need to ask the nerve to the transplanted neuron to work harder, to grow axon faster. Because otherwise, if they cannot make it to their target within certain time, they maybe get tired, or in what we learned, after certain time, they do not make their target, the neuron may be eliminated or something like that. So I submitted an NIH grant for that last week or the week before, try to make the axon grow faster to achieve what you try to say. The second question, I joked, I forgot your second question.
Is how come a very young
yeah,
embryonic
the
cells
the
find
young
their
cell.
ways in the mature brain?
Yeah, you hit the point. The young cells. If they are not young, let's say if you take the mouse, adult mouse, 2 months older, and you make injury, those neurons have a hard time to regenerate. I think many of the people here in San Diego and also elsewhere, if you turn the cells around, make them feel young, change SOX3 or some other kind of a gene, mTOR, and then when the cells feel young, they are young embryonic type of cell, they actually can regrow, right? There are plenty of evidences there, even in vivo. And so I try to make analogy with this type of observation that the neuron we transplanted are young. We usually just use progenitor cell right before they become post-mitotic neuron. And we actually did some experiment, but we never published it because before transplantation, we make the cells getting out of cell cycle and then transplant. They never make it because one is they do not survive well during the transplantation. Two is that when they are post-mitotic, they feel they are old like me, and then they just do not grow. And so that's where I think the key word is when they are young, they can actually grow, they can still find their target and their partners. That's what I understand.
All right. There are some questions online that are kind of I'll combine a few of them. They're on the same theme of degree of maturity of the cells when you do the transplant. So one of the questioners was going, have you ever taken these cells and put them in an embryonic brain to see what they do? And related to that, have you ever really gone exceedingly immature, like taken neural progenitors, not dopaminergic progenitors, but neural progenitors, and seen whether the in vivo environment can provide the necessary cues?
Yeah, very good point. I think for putting the cell into the developing brain, from the beginning, I mentioned to Evan that I use Evan's method, transplant to the developing brain, particularly newborn brain, and that they can incorporate into many areas, many parts of the brain. And remember Steve Goldman's work when he transplanted the cell into the newborn mice, and the cell incorporated into the large area of the brain, replace much part of the glial cells. Now, in terms of earlier stages versus the dopaminergic progenitor cells, so that's a really important question because if we take just the so-called neural progenitor cell that they are not specified or committed to certain lineage, then we may run into an issue that their fate is not determined and only determined by the environment. So it's a kind of area that we cannot predict where they go. And so that's probably not the way we want to intend. And so that is why we usually try to restrict the cell to the dopaminergic lineage, midbrain, ventral midbrain type of, and even further down a little bit to the dopaminergic fit. Because floor plate, ventral floor plate progenitor cell still can give rise to different type of cells. And so this way we can predict what's going to happen. And that I listed in my slide, but I didn't talk about it, meaning what we observed is that when we look at the in vitro properties of those neurons, we have, let's say, 50% of the dopaminergic neuron in vitro. And when we look at the in vivo after transplantation, and quantify them, and it's the same proportion, meaning you actually can predict what you have in vitro for the outcome in vivo. And that is very important. So that's why I think the question hit at the right point, that you need to have the right type of cells for transplantation. I think they're like.
Yeah, so I'm hitting back again on the site of transplantation, despite Alison and Dan asking that question. So one thing I'm confused is, since the damage is mostly in substantia nigra in patients, there's a lot of inflammation, fibrosis, and things going on. And your data suggests that it is better to put in substantia nigra based on the data. But in the striatum, on the contrary, your behavior shows overcorrection, which potentially is high dopamine, unwanted dopamine corrections or more, which we are trying to address with respect to L-dopa therapy and all. So how do you reconcile? What is the consensus? Where we should put the cells, and what's the middle ground or the right approach here?
Very good point. Very good. So what we did by showing the transplantation into the nigra versus the striatum is to show the behavior of the cells, because it's just a model system. When you transplant to the striatum, that model tells you whether the cell produces dopamine or not. That's all. It doesn't tell you much more. So that's why we transplant to the striatum. It will tell you how they grow, how they receive inputs, these type of things. But when it comes to clinics, that's separate. I think we already touched that for now, or even in the past, with fetal tissue transplantation, for example, or for all the clinical trial we are doing, including my own group, we try to do is to put the cell into the striatum. Partly because what you just said. For the most part, I think we want to overcome the distance barrier, because if we put the cells into the substantia nigra, the cell need to go for, several millimeters in order to get to the striatum. And that takes many years. And you may not see outcome in many years. So I think there are issues surrounding the clinical application. I think you are right. And another part, I think it's related to what Dan and others mentioned, that the pathology in that environment, it may also help by putting into the striatum.
Yeah. So related, these projections are great. They need to connect to the right synapses, right corresponding cells. But exuberant connections with unwanted connections when they project too much, that also is an issue. So is there anybody addressing this? Do you see any side unwanted behavior or things like that because of this unwanted connection or unwanted
Yeah, it's a good question. I think we discussed this issue in our consortium. So the animal model we used, like rodents, we mostly look at the motor function, and not many other functions we look at it. I think in the monkeys, we look at many other behaviors. We videotape them and analyze them. And so, like many Parkinson's patients, when you have this chronic debilitating disease, many patients are depressed, have all this kind of related behavior. The monkeys do have this type of behavior, depression, even self-injury, this type of behavior. And when we transplant, after transplantation, even this kind of behavior improved. It's striking, because we normally felt that the cells we transplant mostly deal with motor kind of symptoms. But again, I mentioned that the dopamine neurons, there are another group of dopamine neurons that are related to this rewarding mood, this kind of behavior. And so our cell usually include both type of dopamine neurons. Of course, there are also possibility because their improvement in motor function can also improve the mood kind of symptoms. But in the monkey, we did look for potential unwanted behavior. So far, based on our limited analysis, we have not found such kind of things. And so that makes me feel more confident that this kind of therapy may be quite safe, just like what has been done in the fetal tissue transplantation. You can imagine, you put the whole chunk of tissue, so to speak. So I think it's reasonably safe. But you raised a very good point. We need to pay attention to this type of things,
We're starting to run out of time. I'll take a few of the online questions, and then we'll finish up with the in-house questions here. One of the questions is actually somewhat related to what you just answered. The questioner wanted to know, have you looked at other pathways in your animals other than just dopaminergic, but also serotonergic and cholinergic?
I think it's a good question because those neurons may be also involved in, I think you just mentioned the wanted and the unwanted behavior. And this is almost a routine thing for us to look for other type of cells, including serotonergic and cholinergic neurons in our cells. And we usually observe a very small number of serotonergic neurons. Usually the number is very small. And so that's why we feel that the donor cell, the quality and the consistency of the donor cell is very important indeed.
Okay. Another quick question is, do you anticipate there being any safety concerns with autografts versus allogenic grafts? Or do you think the safety concerns will be the same, or will they be different?
I think for the most part, they are similar in terms of safety, because I think, as Alison mentioned, that we need to make sure that they do not end up with producing other type of unwanted cells, right? But in terms of other type of safety issue, I think auto versus allogenic is probably related to the clinical side. I think, because in clinic, you need to do immune suppression for allogeneic transplantation, for example. And then you will end up with some kind of clinical related issue with immune suppression and so on and so forth.
Okay. Relevant to that, this will be the last online question I'll pitch. The questioner wondered, given your concern for an immunogenic response, have you ever thought about doing co-transplants, for example, with mesenchymal stem cells in your cells to, diminish inflammation or immune response?
It's a good question. I didn't think of that. So the issue will come that how to do the co-transplantation. Probably do not transplant to the brain, right? We don't want mesenchymal cells in the brain in that area. But the co-transplantation to regulate the immune system, it's potential option, I think, to go along if they can regulate the immunological issues.
Great. Those are the last questions online. Any last questions from the audience? Okay.
First of all, thank you so much for the talk. It was very clear. The question I have is regarding to the sex of the monkey, if you see any differences based off of the hormonal changes that are occurring.
Oh, that's a good question. I think in these animals, we didn't look at this issue. We tried to do more work to look at because we usually use all the sexes. We didn't see clear difference, but it's a good question, yeah.
Any last questions in the audience? Okay, I guess with that, we'll come to adjournment of this July installment of the Southern California Stem Cell Consortium. Take care, everyone.
Thank you.